We are going to hear another approach to prevent HIV infection from Dr. Wagner of GeneArt.
Okay, first of all, I would like to thank you for having the opportunity to present our data here. My topic here is programming HIV vaccines, and I'll guide you a bit back to molecular biology first. In the second part, to more immunology and animal studies and clinical trials. Very brief introduction regarding current vaccine strategies. Very short for the sake of time. We have, as you know, we do not have to deal with a single virus. We have different clades of the virus, named A, B, C, D, E, F, G, and so forth. We are not yet sure how we really can target the virus. What are the real correlates of a protective immunity? There's neutralizing antibodies. Of course, if we would know how to induce broadly cross-neutralizing antibodies. Of course, as we've heard, we would like to induce a mucosal immunity. T-cell responses, both CD4, CD8 positive T-cell responses would be highly desirable. Those T-cell responses, again, like the antibody responses, should be broad and should be cross-clade, targeting different clades of the virus. They should be of a high quality, recognize highly conserved epitopes. They should be polyfunctional. And what a vaccine should achieve, it should induce long-term memory. We have altogether 9 open reading frames in the virus, encoding 9 different genes. Envelope for inducing antibodies, GAG, POL, NEF, and a few other regulatory open reading frames are candidates for inducing T-cell responses. We also have quite a bunch of delivery systems, starting from short peptides, recombinant proteins, DNA as the, let's say, the minimal entity mimicking a viral replication. We have a bunch of viral vectors, recombinant adenoviruses, poxviral vectors, and so forth. We have bacterial vectors. We can produce pseudovariants. As I said, a whole bunch, a battery of delivery systems. And finally, once we have our candidate genes within these different vectors, we have to pull them through different preclinical and clinical trials. So this is obviously a tremendous task. Summarized here, the vaccine should be safe, should focus on a relevant virus strain or relevant virus strain, for example, in a given geographic region, should induce T-cell responses primarily towards GAG, POL, and conserved regions within the envelope protein. And the vaccine should, in order to induce T-cell responses, mimic the viral replication. For example, as I'll speak later about, by using DNA vaccination as one of the methods. We have a couple of limitations, just 2 of them highlighted here. If we mimic HIV replication by whatsoever viral vector system or non-viral vector system, this may be linked or is linked to safety concerns. And in order to avoid the safety concerns, we have to try to better understand the regulation of late lentiviral gene expression, which so far is poorly understood. Now, why is the late lentiviral gene expression important to understand here? It's a short summary of the state-of-the-art knowledge. There is a single promoter, enhancer unit, long terminal repeat. From this long terminal repeat, there is a single more than 9 kb message, which is spliced, either multiply spliced or singly spliced, to encode those altogether 9 proteins. Now, if you look early after infection, for example, in cell culture for viral RNAs, you primarily find the multiply spliced, these short RNAs, in absence of any regulatory proteins. These regulatory proteins, for example, REF and TAT, shuttle back to the nucleus. They enhance TAT by binding to an RNA element, the processivity of the RNA polymerase, increasing the levels of unspliced and singly spliced RNAs, which then, by binding of REF to the REF-responsive element, together with cellular proteins, are efficiently shuttled from the nucleus to the cytoplasm. And these singly spliced and unspliced RNAs, those encode GAG, POL, and the envelope proteins. Therefore, our intention was to better understand the molecular mechanisms that capture the late lentiviral RNAs, encoding those important proteins in the nucleus. There have been candidates which were thought to be responsible for the capturing of the unspliced and singly spliced RNAs within the nucleus, and those were either combined action of strong splice donor, the major splice donor site at the 5' part of the RNA together with the weak splice acceptor, or in combination inhibitory sequence elements, AU-rich sequence elements, which somehow contribute to the capturing of these RNAs in the nucleus. Now, the major objective was therefore to assess and to better understand the critical contribution of these regulatory elements, the splice donor, splice acceptor, and the inhibitory sequence elements regarding the expression, the regulation, timely regulation of the expression of late lentiviral genes. The method we have applied was to reprogram heterologous reporter RNAs to mimic HIV post-transcriptional regulation by rational design, for example, by incorporating crucial cis-acting sequence elements. And we did that by enforcing the HIV REF-dependent expression of a GFP protein, as I'll show in a second. The final goal is to provide rationales for the construction of safe and effective vaccines expressing those late lentiviral genes encoding GAG and POL and NEF. Now, how did we do that? We had as a reference the humanized GFP open reading frame. As the codon usage used by the virus totally deviates from the codon usage that you find in frequently expressed mammalian genes, we were sort of constructing a what we called HIF-GFP gene, where we have increased the content of the AU content within the gene by making it look similar to an HIV message. And then in the next step, we were adding either the 5' untranslated region comprising a major splice donor site, or the 3' ref-responsive element where the ref protein, the shuttle protein, binds to together with the weak splice acceptor site. Again here, you see a comparison of the humanized GFP protein in the upper line and the what we called HIF-GFP with C's and G's substituted by A's and T's wherever possible without, as I said, altering the polypeptide sequence. And by doing so, the original AT content of the humanized GFP, which was 37%, was increased to almost 70% in case of the HIF-GFP, yielding finally the totally homologous protein. Now, those were the reporter constructs, the humanized GFP, 5' splice donor added, 3' ref-responsive element added, both elements added, and the same systematic here applied also to what we called HIF-GFP. First of all, to exclude any impact of the codon usage modification on the translation, we have in vitro transcribed these RNAs, normalized for the RNAs, and then in vitro translated these RNAs, and you can see, irrespective of whether it was the humanized gene or the HIF-adapted GFP gene, whether or not there was any cis-acting sequence element added 5' or 3', we ended up with almost identical levels of GFP protein translated in vitro. However, upon transfection of these reporter constructs under control of a cytomegalovirus promoter in 293 T cells, you see that the humanized GFP, not unexpectedly, nicely expressed, again, independent of any cis-acting flanking sequence elements, whereas the HIF-GFP construct didn't express at all. Expression of these HIF-GFP constructs required the co-transfection of the ref protein. Remember the shuttle protein, which is binding to the ref-responsive element. And you see here, in the case of the HIF-GFP, when flanked by the 5' splice donor site here in the untranslated region, and the ref-responsive element as the target sequence for binding of the ref protein, only in that particular case we were able to prove GFP expression. And the nice thing is here that we indeed succeeded to mimic, based on the GFP reporter gene, to mimic late lentiviral regulation in a sense that when we were providing the cis-acting elements in trans, and adapting the GFP gene to the HAV codon usage, then co-expressing the REF protein, we ended up with these nice green cells here. Again, this was to show that the expression is really dependent on a functional REF protein. You see here again, humanized GFP expresses nicely irrespective of any cis-acting sequence elements. Here the HAV adapted GFP gene required co-transfection of a REF encoding plasmid. GFP expression could also be activated by transfecting a plasmid which contains the complete HAV genome. So REF is expressed here from the HAV genome, and by co-transfecting a REF M10 mutant, which is a defective mutant which blocks the cooperative binding of the REF protein to the REF responsive element via Polish expression. So all looks like it should look in the case of the viral replication. Here we were showing that leptomycin B is a drug that negatively interferes with the binding of the viral REF protein to CREM1 X protein, thus preventing shuttling of the RNA from the nucleus to the cytoplasm. That the addition of the leptomycin B sort of decreases the expression of the HIF-GFP reporter. That is also known already from the viral situation that leptomycin B disrupts the interaction of REF with CREM1 X protein. So also here we could show that this HIF-GFP reporter RNA is exported through the very same pathway from the nucleus to the cytoplasm as the viral RNAs do. And this is very similar to the pathway that is used by small nuclear RNAs. This is not the regular export pathway. Now, when we looked a bit deeper on RNA level, you again see that in case of the humanized GFP gene, irrespective of flanking elements, we have the RNA in the nucleus. We have it also when we co-transfect the REF protein, and we have it irrespective of a co-transfected REF construct in the cytoplasm. So obviously, in absence or presence of the REF protein, these RNAs are readily exported from the nucleus to the cytoplasm. Quite different situation in the case of the HIF-GFP construct. We see minor amounts of the RNA in the nucleus. We do not see in the absence of the REF protein significant amounts of the RNA in the cytoplasm. However, when we co-express the REF protein, we see very efficient shuttling of the RNA from the nucleus to the cytoplasm. So once more, we could mimic what we already knew from the viral situation. Now, the situation here summarized as follows. We see that the GFP gene, when adapted, the GFP RNA, when adapted to the codon usage as used by the virus itself, is very unstable in the nucleus and rapidly degraded. Only when we add the 5' untranslated sequence, including the splice donor site, the RNA is stabilized in the nucleus and is only then readily exported from the nucleus to the cytoplasm when we add the REF protein. And as we already know, the humanized GFP RNA obviously is readily transcribed in the nucleus, is stable in the nucleus, and is exported from the nucleus to the cytoplasm. So this is a nice system which is also patented now, which is being used to screen for drugs, preventing REF-dependent export of these RNAs from the nucleus, and is also being used to further look into interaction of viral and cellular factors to transport viral RNAs into the cytoplasm. There is also a lesson learned for the viral situation. Now, we could show here, and for reasons of time, I'm not going to go into detail. This is work, by the way, done by Markus Graf, who's also here in the audience during his PhD thesis. The very same situation applies for the late lentiviral gene. Same situation here. The GAG message is rapidly degraded in the absence of the 5' splice donor site, stabilized with the 5' splice donor site, and readily exported with the REF-RE system. But the lesson learned is here when we were using a mammalian codon usage for the GAG gene, for the viral GAG gene. It is like the GFP gene. This is readily transcribed in the nucleus. and it is readily exported from the nucleus to the cytoplasm. So this seems to heavily simplify the situation if you want to go for DNA vaccines, for example, that are encoded by a plasmid or delivered by a DNA vaccine. So practical implications here for the HAV candidate vaccines is if we RNA encode and optimize late lentiviral genes, we facilitate, as I've shown, the expression of GAG or POL or also envelope. This is not due by enhancing the translation. This is rather due by stabilizing the RNA and supporting the export of the RNA from the nucleus to the cytoplasm. We get rid of this complex viral regulation down here, get rid of the REF-RE system. We can avoid also the 5' splice donor site with the packaging signal. This has a few implications. Hopefully, as I show, we will reduce the risk for recombination and RNA packaging. This will be associated with safety, have safety implications, and hopefully also translate into immunogenicity. Implications on safety, a reporter system that we have established, a proviral construct, a plasmid containing more or less the complete HAV genome with a few deletions. So if you transfect this plasmid into 293 T cells, we'll not end up with replicating particles. We have co-transfected with this partially deleted viral genome a wild-type packaging construct encoding GAG and POL. And in case of a recombination event between the packaging construct and the partially deleted viral genome, in case of a recombination event, we would see virus-like particles being released from these cells. And if we add these virus-like particles on an indicator cell line, which encodes the secreted alkaline phosphatase under control of an HAV promoter, we would expect viral replication and thus increase of activity of the secretory alkaline phosphatase. And exactly this is what happened. If we did the very same experiment with the RNA encodon optimized GAG-POL construct, as you can see here, we have an initial blip of activity of the secretory alkaline phosphatase, which, however, as the cells divide and as the virus does not replicate, goes down to background levels. So no generation of replication, competent recombinants when we were using those codon-optimized constructs. In another experiment, we were sort of enforcing recombination by using an infectious molecular clone of the virus and substituting parts of the GAG and POL gene of the wild-type GAG and POL gene by the RNA codon-optimized gene here, by the complete GAG-POL gene, and by certain parts of that. And also to summarize that very briefly, as you can see here, when we were transfecting the parental molecular clone of the virus, you see after a couple of days in a permissive MT4, CD4 positive T cell line, you can see here easily nice replication of the virus over time. You can also see that 3 out of 4 variants that we have been generating do not replicate at all in this system. So just by substituting the wild-type sequences by the codon-optimized sequences, we were able to abolish replication. There was only one mutant which was replicating with a significant delay and also to minor levels of infectious virus. When we were using the RNA encodon-optimized GAG gene, again putting it under control of a cytomegalovirus promoter and either injecting it into mice by intramuscular immunization or by administering it through a particle gun after coupling of the DNA to small gold particles, we found that the synthetic GAG gene, the codon-optimized GAG gene, in all cases was by far superior to the wild-type sequence, irrespective of whether we're using the particle gun or the intramuscular immunization, and also irrespective from the fact whether or not we looked for antibody titers, for secretion of cytokines, or for cytolytic activity in that mouse model. Now, we did not want to develop vaccines for mice, rather for clinical trials to be performed also in developing countries. Therefore, we did in parallel analysis and extensive over more than 10 years molecular epidemiology survey with collaborators here in China. We were able also to clone a virtually full-length clone of a virus strain, which is believed to be a representative virus strain for India and China. Based on these sequences, we were picking out the GAG open reading frame, the POL envelope, and part of the NEF open reading frame. We were together with GNARD, RNA, and codon optimizing these open reading frames. GAG, POL, and NEF were sort of summarized to a 160 kilodalton artificial read-through protein plus envelope. Based on these optimized constructs, DNA vaccines were established, as well as a pox viral vector by Mariano Esteban together with Sanofi Pasteur. These candidate vaccines were available as GMP materials and also used in clinical trials, as I'll show in a second. Just a brief comment about optimization of the sequences. Codon usage has been adapted. The GC content has been increased. RNA secondary structures have been removed. Cryptical and active splice sites have been removed. Polyadenylation, IURH sequence elements have been removed. Sequence repetitions have been removed as well. Just to name a few parameters that were applied. Well, these were the genes that have been used for the generation of the vaccines. Now, the study design of a second phase 1 clinical trial was as follows. One group received a DNA vaccine encoding the GAG, POL, and NEF, plus the envelope open reading frame, 2 injections at week 0 and after 4 weeks, and at week 20 and at week 24, 2 booster immunizations using a NIVAC virus. NIVAC stands for New York Vaccinia virus. The New York Vaccinia virus is based on the Copenhagen strain of the virus, which has been used also to eradicate the pox viral infections. This is a highly attenuated virus, which is able to replicate only a single round in human cells. So this was the prime boost scheme, and that has been compared to an immunization with a viral vector alone. Studies have been conducted in Lausanne, 2 times 20 HIV negative volunteers, and several immune parameters like interferon gamma and IL-2 producing T cells by an ELISpot analysis have been determined. FACS analysis has been used. Functions of T cells have been analyzed, and epitopes have been mapped. This was a collaboration with 2 clinical groups, Giuseppe Pantaleo in Lausanne and Jonathan Weber at the MRC in London. Just to summarize the results here on one or two slides, the take-home message is that the DNA prime NIVAC booster immunization was by far superior to the NIVAC immunization alone in terms of the response rate. Almost 100% of the individuals, vaccinated individuals in this group here, responded as we had hoped to see to the vaccination, and these responses also have been long-lasting over more than a year. One important message also was that the magnitude of the T-cell responses in the prime boost was by far superior to the magnitude of the T-cell responses that we have measured in the group that received immunization with the viral vector alone. And another important take-home message is that both the CD4 cells and CD8 cells were polyfunctional T cells, polyfunctional in the terms of the production of IL-2, gamma interferon, and TNF-alpha. This polyfunctionality from studies in long-term, non-progressing HIV-infected individuals has been correlated with a mild disease progression. So we were quite happy to see this polyfunctionality within the T-cell responses. I would like to sum up here. I demonstrated evidence that we better understood the critical contribution of the impact of the splice donor and splice acceptor site and the inhibitory sequence elements based on a heterologous HIV reporter sequence. We were able to confirm the molecular mechanisms and their functions regarding the export and the expression of latent lentiviral genes. We were also able to bypass the concerted action of these cis-acting sequence elements by extensive codon usage modification in the late lentiviral genes. We could also show that we were able to attenuate the viral replication by codon usage modification, and taken together, this was the basis for the construction of safe and immunogenic HIV candidate vaccines, which are able to mimic immune profiles as we see them also in long-term non-progressing individuals. I do not want to conclude without mentioning that RNA and codon-optimized genes are more and more frequently used in the design of recombinant vaccines, of therapeutic antibodies. Here is a long list of immunogens that have been optimized by GNERD for several customers, viral antigens, bacterial antigens, and so forth. This technology, RNA codon optimization, is not limited only towards improving the expression of bacterial or viral or parasite immunogens. You can find at the QIAGEN web page, GeneGlobe web page, essentially the whole mammalian, human, and murine genome, genes adapted for high-level expression in mammalian cells as well as in E. coli. And GNERD also significantly contributed towards completing the mammalian gene correction, a major project driven by the National Institutes of Health here in the United States. And GNERD was asked like 2 years ago to help completing the mammalian gene collection by synthesizing all these genes that were leftovers based on conventional technology. So there was a bunch of genes that was not able to be amplified by PCR, by conventional cDNA cloning strategies. Those were the difficult ones. Extra long, high GC content, hairpin structures, direct repeats, and GNERD luckily was able to provide the NIH with these genes. So there's lots of application for gene synthesis. Well, I would like to thank the people in my university group for their contributions. I've mentioned Marcus Graf doing a lot of the molecular biology analysis that I've presented. I'd also like to thank my colleagues at GNERD here, some of the important people listed. Thanks.